Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Andrew Lowell from KBI Biopharma, Inc.
Hi, Leah. Thanks for introducing me. Hey, everyone. Today I was hoping to go through the slides here and give you a little view into our implementation of single-use fermentation technology here at KBI Biopharma. So thanks for taking the time, and I'll go ahead and get started. My site specifically is located in Boulder, Colorado. The agenda for today, I was hoping to give you a quick overview of KBI, what we do, and kind of the sites we have globally. The most important part here is, what were our drivers and strategy for the single-use fermentation implementation. It's, I wouldn't say it was an easy decision for us. It took a lot of deliberation, but we are happy with the outcome. I'll go into a little bit of how we couple our process design to the boundaries of our at-scale single-use fermenters. We think this is a kind of a new approach to process development and a non-traditional approach. And finally, I'd like to get into a little bit of the challenges and, of course, the successes of the single-use fermentation systems here at KBI. Just as a kind of headline here, our process design to the stuff equipment is enabling us, currently around 6 to 7 grams per liter for protein titer, as well as, cell densities somewhere around upwards of 200 OD. So KBI, like I mentioned, we are a global contract manufacturing and research organization. These are North America locations that we do have. Like I said, I am based in the Boulder, Colorado location. At this location specifically, we do microbial work that includes everything from strain development and cell line development to process and analytical development. We do have clinical and commercial cGMP manufacturing suites on site, which our processes end up in as we move through development. We also have all the associated departments, to support that commercial and clinical facility in terms of QC, analytical, formulation, and stability. We also have a particle characterization core group on site as well. Some of the other sites, real quick, also in Colorado, we have acquired Elion Labs in 2018. They do our analytical work for us and lead the way in biophysical characterization. San Diego, we have Alliance Protein Labs for analytical technologies. We also have a site in the Woodlands for cell therapy in Texas, also including process and analytical development. They also have GMP manufacturing and testing there. Our mothership, or kind of the original location for KBI, is in North Carolina. They are focused on mammalian development and GMP manufacture. They also have some of the similar departments as we do in terms of support and analytical QC. They also have a mass spec core facility and focus on cell-based assays as well. There is another site in the Research Triangle Park as well, focusing on cell line development, process development, and process characterization. We do have 2 additional sites in Europe. We have KBI Leuven in Belgium, They are responsible for analytical work as well as formulation and QC services for both non-GMP and GMP product testing. We also have Solexis, which is in Geneva, Switzerland. We acquired Solexis as an affiliate of KBI in 2017. They are the leader in cell line development and candidate selection technologies as well. Moving a little more acutely to what we do at the Colorado site, as most of my discussion will focus around our operations here. Like I mentioned, we do process and analytical development for first-in-human and clinical programs. We also focus on late-stage commercialization, including process characterization, process validation. So we do a range of development and GMP manufacture, all the way from first-in-human, early-phase programs to commercial production of drug substance here on site. We do have two manufacturing streams within the facility. We have a larger, one of our larger trains at 1,500 liters, and that is a stainless steel system. And then, more focused on this presentation, in the last 2 years, we have built a 300-liter single-use production suite. And I'll, of course, get into more detail on that as we move through these slides. We, again, have the supporting departments, as I mentioned before, in terms of quality assurance and quality control, validation, operations. We have a larger kind of newly formed MS&T group that we continue to add responsibilities and capabilities to. Then, of course, we have an engineering department here on site as well. So getting an even closer look at, what we do here and what my group does specifically, as I mentioned, I'm the Associate Director of the Upstream Process Development Group. Within our microbial process development work stream, we have a couple of trains that we use, as a jumping off point for all projects that come into our lab. We tend to use our small scale single use system, and that's our Amber 250 for screening, optimization, but then also late stage PC characterization, DOE design, as well as small scale model qual. So every project usually starts there and ends there through its life cycle. From that small scale single use system, we can either move into our 10-liter scale, which, if we're moving in that direction, we most likely are going into our larger scale GMP facility, which is our 1,500-liter suite. But then the other way that we can move is also under our 30-liter scale. This is using our single-use thermoscientific high Biopharma single-use fermenter. We use this scale for harvest development and also scale-up confirmation as well. And then, of course, from there, we move into our 300-liter suite at a GMP scale for manufacture, and that is also a 300-liter high Biopharma single-use fermenter as well. And this scale, the 250 mil, the 30-liter, 300-liter for single-use, is a system of development and train that is needed, and we have comparable systems across that train. I should mention, as I said before, the 300-liter suite is I'd say about 2 years old now, and I will get into what we've run in that suite and the performance of those projects historically. So the big question is, or decision, I guess, is why did we decide to implement single-use fermentation technology here at KBI Boulder? Traditionally, as a stainless suite, people sometimes stay within that realm, and the single-use fermentation technology at the time that we made this decision, which was probably about 2.5 years ago, was new to the industry for fermentation, but we had enough experience with staff, with mammalian cell culture in the past, that we felt confident we could implement single-use technology here on site for many of our clients. So the driver, the first driver was that, you know, we, as a contract research and manufacturing organization, we have clients that come to us with early phase programs with a relatively small preclinical and clinical material need. You know, certainly the 1,500 liter stainless system would give them more material or drug substance than they would ever need. So it was a little bit overkill. So we were looking for something that we could produce kind of the material quantities that they needed without overdoing it and just having to waste a lot of the material we produced. One of the other main drivers was we get quicker process design to GMP production, you know, through our platforming and equipment-based development. And certainly that is extremely attractive to clients as well, with a tighter development timeline, or not a tighter, but a quicker development timeline. We do get a higher throughput through GMP than pilot suites, resulting in more flexibility in the schedule. I'll go into this again in the slides coming up, but you know, obviously with single use, the resources that are needed to operate it, as well as you know, excluding sterilization and cleaning procedures and validation procedures and testing that encompass, you know, traditional stainless systems. We get to see those advantages in throughput in our suites and our labs, and kind of offering that flexibility to the schedule. As I just mentioned, the validation requirements are much less with our single-use technology at the GMP scale. This saves you know, countless numbers of hours, you know, throughout the department. So another one as well, you know, decreased cross-contamination between products. You know, we are taking these fermenters and discarding the fermenter and pulling the bag out. We don't have to worry about cross-contamination of products, you know, between campaigns. And like I said, so, you know, the ease of operational requirements equate to huge time and cost savings. I like to say that, you know, fermentation in general is about 25% of the actual fermentation, and 75% is custodial, cleaning, sterilizing, solution prep, things like that. So cutting out the cleaning and sterilizing certainly gives us more free time to either increase our run rates or focus on surrounding operations around the fermentation. So in summary, you know, there's a greater flexibility for KBI as a CMO organization for these rapid execution of projects. You know, we have significantly increased our run rate using single-use technology, and again, I'll get into that as we move forward here in the slides. So these are the two systems, the Thermo Scientific High Performa single-use fermenters that we have. There is a 30-liter pilot system. This resides in our Upstream process development laboratory. You know, this is a non-GMP laboratory. Like I said, this is a process development laboratory. So, you know, it's not as, I guess, complex as the 300-liter stuff in the GMP suite. What you can't see in this picture, and again, this is this unit here, you can't see, you know, where the 300-liter has an HMI for their TrueBio controlling software. Out of the picture in this unit, we have a computer that also runs the same software. You know, Coming from a stainless world, this was, I would say, a little bit of a breath of fresh air in terms of, getting used to operations with the single-use technology in the process development lab. This includes, the weldable connections that we found to be pretty quick and easy. And like I said, the less preparation for fermentations in terms of sterilization or pre-use cleaning and breakdown. So we can perform multiple fermentations in a week, where traditionally that time would be taken up with operations around the stainless steel fermentation preparations. So again, the 300-liter system is here. It just kind of looks, it's a big brother to its 30-liter. This is in our GMP small-scale suite, and I'll get into kind of what we have run in that 300-liter system a little later in the slides here. So the strategy for our single-use fermentation implementation, this begins as early as cell line development. Understanding the boundaries of at-scale 300-liter self-performance really drives how we begin our development path at the beginning. So our cell line screening and initial evaluation is done in our 250 ml single-use fermenters. And then platforming and optimization of the fermentation with an equipment-based process design follows that. Traditionally, with stainless systems, the sky was the limit in terms of development. There was rarely processes that came through that would not be able to fit into a stainless system, or not, the stainless system could not deliver the OUR demands or oxygen demands of the culture. So this is, when I talk about a equipment-based process design, this is kind of what I speak of here. So the 30-liter high performer self process demonstration and confirmation of design is what follows from our 250 milliliter single-use fermenters. So, really the limitations of the single-use fermentation systems dictate our design approach. As I mentioned before, the process design with stainless systems was more or less limitless, in terms of variation, OUR potential, and growth strategies. We all know that with microbial fermentations, there are high growth demands within these fermentations. There's high oxygen mass transfer rates that are necessary for most of these. With the single-use fermentation, the lack of a pressurized system limits this OUR potential. Traditionally, process development has been aligned to scalability in stainless systems. So, our at-scale stuff now defines the boundaries of platform process development approach. And the two are interconnected, where, there is parallelized development designed with at-scale constraints. So I'll go into that more in these next slides, where I talk about the platforming here. To combat the limits in OUR potential, we really have platformed our process development within the Upstream development laboratory. We have platformed the batch phase for most of these projects that come in. And this is to support optimal growth, matching the culture demand. You can see within these trends, the batch phase of the culture is here, and then we begin feeding. So, our batch media is designed optimally to meet the needs of the culture. and give us the optimal health and cell density during this phase. And then really, once we get into a fed batch mode, we feed both, and we limit the feed rate as well as the nutrients to the culture to match demands, and we move that along, and that is what we refer to as our early growth phase. And then really, the critical point that we found with all these single-use fermentations is our late growth phase, right? As you're, entering your late growth phase right before your production phase or induction, or that is the riskiest zone in terms of not being able to deliver as much oxygen as you need to the culture. So to combat that, we tend to go into a ramped feed rate to get our culture where it needs to be from a cell density perspective. And then, at this point, one of the methodologies we're using, we can decrease the temperature both to decrease metabolism of the culture, but also to improve our OUR capacity. With this, we then enter a linear feed to maintain the OUR somewhere below 300, as you can see here. And really, we hold that until we reach the proper cell densities for induction, or what have you, what may be your production trigger. So again, here we have a figure with a full fermentation. Like I said, feed rate in growth phase is kind of optimized to maximize culture growth while also limiting OUR. You can see here, this is the OUR, the blue line here. We get up to about 275, and that typically is the limit we try and design around based on our capacity at the 300-liter and 30-liter single-use fermenter. But we also go through a series of temperature drops to limit that OUR. Here's the first time you're seeing the production phase as well, where we drop the temp even more and leave that temp constant throughout. And as we all know, that during production phase, we usually do see a decrease in our OUR as we proceed to the end of fermentation. So really, what this platforming of our process design for the single-use fermenter does is it allows us to focus on the production phase. We've platformed the early and late phase growth for these projects or for these different cell lines that come in. So we then, can focus our efforts on the production phase and increasing our titer and improving product quality from that point. So in this figure, I'd like to show you a little bit of how we compare from our 30-liter to our 300-liter fermentation system. Again, as I mentioned, both units are Thermo Fisher Scientific High Performa SUFs, or single-use fermenters. Excuse me. We do see slightly better aeration in our 300-liter unit. Between the two of them, you don't really see an OUR here, only because off-gas analysis, we do not have that in the 300-liter suite. We tend to focus process development around the exhaust gas, and then it carries over to 300-liter at the same level. So, you see here as a max O2 pull, the 30-liter is a little less efficient at mixing and aeration and OTR. The cool down times, again, 300-liter, at least in our suite, we have a glycol-fed unit in the 300-liter system and an air-cooled unit in the 30-liter. So that's what this difference is. We just get more efficient cooling at the 300-liter. The titers are comparable. We've seen this through all projects. As I mentioned, we are seeing upwards of 6 to 7 grams per liter in the lab for this project specifically that's shown here. We saw from 3 to 6 between each system. And then for cell densities, we're seeing anywhere from 130 to 200 OD coming out of these fermentations, and the correlating, 22 to 29% wet cell weights. So we are seeing comparable biomass and product titers at both scales, and we believe it's comparable to stainless steel outputs on some of these processes. So of course, we're very happy with that. So I'd like to mention this slide is just one of the programs that we have put through the lab and the GMP facility. Obviously, I have here on the table the present single-use fermenter max output for our Biopharma unit. Our client would not allow us to show that data, but we can put the numbers in the table here. What I want to highlight with this is kind of through our single-use systems in the Upstream process development laboratory, we do see very close correlation between all the systems, both at 250 milliliter single-use fermenters that we use and the 30-liter single-use, and how that transfers to the 300-liter. We see very similar wet cell weights, final ODs, and as well as titer, most importantly, and product quality as well. So, again, we couldn't show the data or the trending for that client, but presently, our max output on our single-use fermenter system, and that would be our 30-liter system, we are seeing about 29% wet cell weight and final ODs of around 200, and titers just around 6 or a little above. In the amber system, the smaller system, which has a little better oxygen transfer capabilities, we actually have seen upwards of a little over 7 grams per liter titer and ODs up over 220. But for the 30-liter specifically, we're just slightly below that. Just to highlight, kind of since we decided to go down this single-use path, what kind of programs or output have we had at KBI Boulder? Like I said, we've had this single-use fermenter, at least our 30-liter, for about a year and a half now. And in that time, we have put almost 500 250 mil single-use fermentations. We've done 30-liter single-use fermentations with our Biopharma stuff. I should mention all of this is coming from approximately 8 single-use programs through our lab to date. And, from those programs, we've done around 17 300-liter GMP fermentations. So this has obviously been a pretty popular path, both from a client perspective and from KBI's throughput and efficiency perspective. We do have 2 new single-use programs planned in the next month. So, these numbers will continue to grow. We'll continue to get clients with the needs for maybe a smaller amount of clinical material and a higher throughput, both through the process development laboratory and the GMP suites. So all in all, we have had, high throughput and success resulting from platforming our single-use fermentation process design. It does, take a little effort to get out of that stainless steel train of thought with process development, and I think we've been pretty successful at doing that. I guess, if you had mentioned to me previous to actually implementing a single-use program that we would kind of get these throughput results and, successful completion of programs to the lab, I might have been a little skeptical, just from a kind of a fermentation specialist standpoint, but I certainly have been proven wrong in that train of thought. And I would say the majority of the programs now through our process development lab are single-use and are slated for the single-use suite on the site. So, all this isn't, I would say, just rainbows and happiness. There are some challenges that I'd like to bring up, that we're encountering with the single-use. Obviously, the overriding theme here is kind of OUR limitations, right? So we typically see a maximum of 350 to 400 OUR in our 30-liter stuffs through fermentations. But as I mentioned, we're actively mitigating this with the platform process design approach, and we've been successful in that so far. One of the big ones that I think people that are familiar with mammalian cell culture single-use and microbial single-use is managing your supply chain. So, you go from running in, first a development laboratory, you go from using, one single-use bag every 2 to 3 weeks in a mammalian model to 2 to 3 bags per week for a microbial model. So there are challenges in managing your supply chain and keeping on top of orders and making sure everything is stocked and in place for the fermentations. And that kind of leads into the next one as well. We also had to manage kind of the decreased expiry with single-use integrated pH and PO2 probes in the bag. There are certainly options outside of this for Thermo Fisher for their system. You can use reusable probes. That helps. We've decided to kind of move towards a completely single-use design. So we do experience kind of a decreased expiry and with those probes. I also know that Thermo Fisher Scientific is looking into kind of additional vendors and looking to increase those expiry periods. So maybe this won't be such an issue in the future, but this certainly goes into maintaining your stock and maintaining the supply chain and having enough bags for you and making sure your probes aren't entering expiry. One of the ones that this last one I'll mention is one that we've recently encountered, and that is transitioning out of single use back to stainless at scale, given an increased demand from the client or from the project, right? So, single use may have worked for their early phase first in human supplies. As they ramp up their clinical work, often the material demands increase, by a substantial amount. So, as these processors are designed based on the limitation of a single-use system, it's not as easy, I think, as one may think to just jump back into a stainless system. That would require a reboot in both process development work and assessments. So, we are just now getting into this new realm for clients that we've started in single-use systems and now may want more, so that may or may not be a challenge moving forward. The 300-liter system that we have on site, speaking specifically to the GMP1 and suite, we do have a few challenges associated with that. There are some materials of construction that may not be compatible eventually for cleaning chemicals, anodized aluminum in some areas, things like that. There are customized mass flow controllers that at times prove difficult in calibration practices. I think we would also prefer that a single-use option for an exhaust gas manifold was one of the other ones that came up. Security levels within the controller software is something that recently has been brought up as well. And then switching from those traditional reusable probes that I mentioned to kind of disposable probes as we're trying to implement also requires some code changes in the software. The good thing about this is I think Thermo Fisher Scientific has been very attentive to these, and it truly is. I think there's technology improvement opportunities that exist in the relationship between vendor and customer here. So I'm hoping, with that relationship, these matters will become less and less important. So real quick, as a summary here, as I mentioned, we have been successful here at KBI Boulder implementing single-use fermentation systems. They've allowed greater throughput and efficiency in the laboratory, which again leads into accelerated project timelines for our clients to GMP production for the production of their material. And truly, coupling the process design to the stuff, equipment capabilities, it's enabled us to get processes with high cell densities and high titers. And it also allows, rapid scaling among single-use systems. So the platforming truly has allowed us to, standardize our fermentations coming into the single-use system and really focus our process development efforts on the production phase, and have that time saved in the platforming of the batch phase or the growth phase. So truly, we are getting an improved flexibility at KBI as a CMO organization, given the efficiencies, the throughput, the time savings associated with single-use fermentation technology. And this, of course, is necessary when we have to address multiple diverse programs through our single-use systems. We do have, many different processes coming through, and having that ability to platform and move through our development systems quickly is extremely important. Below is just kind of an estimate of what I could put together here, our usual timelines now, and certainly improved over our stainless timelines. But, within about 4 months, we can get a single-use fermentation program process, barring an extremely complex fermentation, we can usually get a process confirmed and locked within upstream process development. I'd also like to acknowledge, again, a lot of the team here at KBI that have helped, and of course, Thermo Fisher Scientific in the support that it took to actually implement these single-use fermentation technologies on site. It being a new venture for us, it certainly made it much smoother to have their support through this. So with that, I guess I'll take any questions that might come through.
Okay, great. So the first question is, in terms of hybrid facility, what features of the single-use fermenter have been most helpful for this type of workflow, and why?
Okay, good question. I would say, the decreased operations around fermentation that I've talked about. I think I mentioned, fermentation as a whole is about 75% custodial and 25% actual fermentation. So, decreasing those resources around those operations in terms of sterilization and cleaning and validation allow us to be a little more flexible with our plant schedule and lab schedule and save time and, again, those resources. So, lack of cleaning validation is also a big one that works in our facility as well, and has proven to be kind of advantageous from a resource perspective and minimizing that need in single use.
Okay. And how important is the choice of controllers on a single-use fermenter? What controller do you use and why?
Yeah, also a good question. So, in general, the hardware for the stuff is going to be your least complex part of the system. I mean, it's a pretty simple system at first glance when you're looking at it in terms of, the jacket and the controller and the pumps, and it's a fairly simple and intuitive system. Um, Really the training and I guess most of the resources go towards the controller and the software around the stuff. And we have the TrueBio software within the unit, and I think that what that gives us is once you become familiar with the system, the amount of flexibility that is available using that software. Again, this used to be the Delta V based system. So in fermentation, we're implementing complex feeding strategies, oxygen delivery systems, things like that, and the TrueBio controller allows us to build these complex strategies around our fermentation. But also allows us to kind of customize, calculations and outputs, right? So OUR, CER, things like that, we can form calculation blocks around those and get that real-time data.
Okay. And how much has throughput increased in the lab with the implementation of single-use technology?
Wow. I would say, at this point, we're almost double the throughput within a week. As I mentioned, all the operations around a stainless system in terms of, empty vessel sterilization, full vessel media sterilization, cleaning, things like that. We get to take most of that out. And it's, as I mentioned, kind of in the challenges of the supply chain is that, our run rate has increased so much. It's a new model to get used to is, keeping those consumables and bags stocked for those increased run rates. So I would say, in a typical week where we may be able to do one, stainless steel fermentation set, maybe a 48-hour fermentation or something of the like, we're now able to do two just because, a lot of the supporting activities are omitted from the single-use systems.
And can you elaborate on the challenges that you face with the single-use system?
Yeah, I mean, I guess as I mentioned before, OUR or limited OTR capabilities is kind of the big one. We mitigated that risk with our platforming model for development, but of course, I keep mentioning it, but I think it's worth mentioning. The supply chain issues are something that need both from a ordering on a process development laboratory scale, but then also from a GMP scale, and how are you going to store that number of consumables, and do you have space for it, and really considering those things before you jump into single-use technologies is, I think, very important. And again, I'll mention it, it's fairly new. I mean, like I said, we're about a year and a half into this from a development and GMP perspective, but, transitioning out of those single-use systems to stainless for an increased material demand or material needs from clients, we're pretty, like I said, we're pretty new into this this problem. I think we have things in place to kind of mitigate, risks around it and build a development program for those specific needs and those specific programs that are transitioning out of single use. But that's certainly one that, I think will be a challenge moving forward for us.
From start to finish, what has been the response from your clients employing single-use fermentation technology and GMP? Are they seeing comparable results in terms of success rates and product quality?
Yeah, so all in all, our clients have, appreciated the flexibility and shortened development timelines with the stuff. I think that's pretty attractive to, early phase FIH programs that are looking to get into trials fairly quickly and move their projects along quickly. The success rate, I would say, has increased due to the limited operations around the fermentations. We can truly focus on the fermentation, process as a whole, as opposed to getting too wrapped up with resources focused on those supporting activities. Currently we're limited by OTR capabilities of the stuff, and we reach a little lower cell densities as compared to our stainless systems. And with those lower cell densities, we see a slight decrease in titer, but no noticeable effects to product quality so far. And I think from the clinical material demands associated with our SUF programs, it's acceptable, and it's an acceptable trade-off for the flexibility and throughput gain from the SUF. So I think even though we may see a little lower titers, a little lower cell densities, I think the flexibility that we achieve for both the client and our facility is certainly worth that.
Yep. Okay. And it looks like we have time for just one more question.
Okay.
If we didn't get to your question, we will send those directly to Andrew, and he can follow up with you. So the last question is, as a CMO, are there any benefits of the single-use fermentation that really stand out as an advantage for your business model?
Absolutely. I think I mentioned in the presentation, flexibility is key as a CMO. Both adapting to changing timelines, changes in the process, and client needs and demands. That happens throughout our business, and it happens quite often. So, we are tasked with delivering results on short timelines. So, of course, the higher run rates help with that and help the client. The stuff improves kind of this high run rate model by allowing us flexibility in terms of scheduling for both, process development and higher throughput than traditional stainless systems. So in terms of flexibility and scheduling, that allows the client to get their project into a GMP space much quicker than we would for traditional stainless systems. I hope that answered the question.
Okay, thanks, Andrew.
Yep.
And thanks to our audience for joining us. The recorded version of this webcast will be made available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We invite you to join us for future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.